Hello everyone, very warm welcome to this Cambridge Science Festival event on health data science and COVID-19. My name is Richard Horton, I'm the Director of Operations for Health Data Research UK Cambridge. So just before we get started on the presentations, a couple of quick housekeeping points. Firstly, just to let everyone know that this event is being recorded. Secondly, if you have any questions, please enter them at any time in the presentations in the Q&A function, which you should see at the bottom of your screen. And we'll work through these questions at the end of the presentations. And finally, there will be a poll at the start and end of the event, and we'd be very grateful if you complete that. So Health Data Research UK, or HDR UK, is the UK's National Institute for Health Data Science. And in this event, we'll have presentations on HDR UK's work nationally on COVID-19, and then 3 short presentations of examples of COVID-19 research being conducted from HDR UK Cambridge. So firstly, it's a great pleasure to introduce Caroline Cake to present on HDR UK's work nationally on COVID-19. Caroline is the CEO of HDR UK. And under her leadership, the Institute, since its inception in 2018, has made great strides. So great pleasure to welcome Caroline, and I'll hand over to Caroline now.
Thank you so much, Richard. And it's great to see you all, see you all virtually this evening, and thank you for joining this discussion on health data research. So if you wouldn't mind going on to the first slide, to view it, would be great. What I'm going to cover today is, as Richard said, HDR UK is the National Institute for Health Data Science. And what that means is what we're doing is bringing together all sorts of data about all of us to actually enable discoveries that improve people's lives, so that we can do great research and make discoveries that are, improve our health, our care in all sorts of different ways. So what I'm going to do is update you to share with you a number of things about what health data research is doing nationally, but also going into then what's happening, what we've done over the last year in COVID. So as this slide here shows, health data research covers a whole range of different things. So and actually it's been around for many, many decades. So we're starting back with a good example from John Snow, whose work back in 1854 on cholera. And he was, when cholera outbreak happened in London, he was the first to discover that it was actually associated with a pump pipe. And this little picture you can see in the top left is showing plots of people suffering from cholera and showing the kind of the outbreaks of it, and was able to identify and pinpoint where the outbreaks were coming from, and then able to address it. And if you look forward in time, I won't go through all of these, but to give you a kind of flavour of the amazing things you can do with health data, you can then see if we go through to the bus conductor survey, which was a really interesting study done in 1953, which is when looking at the work done around the difference between bus conductors and bus drivers, and finding that, and looking at the difference in cardiovascular disease and heart attacks, and finding that the sedentary lifestyle or working style of the bus drivers meant that they had a much higher rate of heart attacks compared to bus conductors, who were walking something like 60 steps a day during the working day on that. So very, very different kind of outcomes for them. And if we kind of wind right forward to more recently, 2017, we've got things like the work done on chest images using artificial intelligence to actually use and actually be able to read images and get kind of results and things from that. So you can see the kind of evolution of technology means there's a lot more data that's come out, but also all sorts of great purposes and uses, and a wide range of different uses of health data research. Next slide. So what we're doing at Health Data Research UK is really 3 broad things. So the first one is bringing together the data, so uniting health data, so we can actually use a wide range of different sources of data. Secondly, then improving that data, so actually making it really useful for different sources of research, and that's both tools and methods, and I'll talk a little bit about hubs as well at working on that. And the third thing is then actually the use itself, so encouraging the variety of research uses that go on, but also building the skills and capabilities to enable that use. And all of this then is enabled by the kind of working very closely with public, patients and practitioners. So really we get a very trustworthy approach to doing this. And then at the heart of all of this, we've got so that we're getting the real impact and benefits for the public and for all of us. So we are, as I mentioned, a national institute. So and this shows the kind of where we're located across the UK, but a wide range of different types of organisations involved in HDR UK. And you can see the organisations doing research work, and we're obviously today talking from the HDR Cambridge site, but also work that's going on in the research hubs and in the training activities that are going on across the country as well. And you can see Cambridge also mentioned in there, and also on the hubs, Gut Reaction is based in Cambridge as well. So it's fantastic to be talking with you today. Next slide, please. Now, when I mentioned that there's all sorts of different sources of data coming together, you can see from this side, these are the sorts of organisations that are involved in that data. So you can see there are NHS hospital trusts, there are organisations that are biobanks or cohorts, there are charities, you'll see in there some familiar logos from those. So all of them have got a wide range of different sources of health data, which is then really useful for research. And the types of research that are happening then across a range of different areas in terms of, we've got in the purple here, the understanding causes of disease, and Ewan and Angela and Adam will be kind of talking through interesting kind of work that they're doing there. Clinical trials work as well, and then data being used for public health, and then also for clinical care. And then across in the green areas here is some of the tools and techniques used to help enable the use of health data, be that applied analytics or artificial intelligence activities, or phenomic work that's going on with the human phenome. And across all of these, then there's a when I'll kind of talk through in the COVID work, you can see applications of these happening that have helped during the in the UK's COVID response. So I mentioned earlier on the hubs. So these are, if the previous slide was talking about the research that's going on using the data, the hubs then are organisations that bring together charities, NHS organisations, industry partners, and academic partners to really then enable the data to be useful for different purposes. And you can see from some of the names here the types of purposes that are going on. So, for example, DigiTrials is working on clinical trials, Gut Reaction is on inflammatory bowel disease, you've got DataCan on cancer, Breathe, which is doing respiratory health, Insight on eye disease, Pioneer on, which is actually on acute care, and then Discover Now is on using data and real world evidence. So there's kind of range of different uses that are happening, which really just shows the diversity of ways in which health data research can be useful for us. Then across this, we have something called the Gateway. This is a little snapshot, and you can go online. You can see there the link there to it. So this is a portal really to those different sources of data. So what it's doing is allowing you to discover what sorts of data there are. And you can see from here, there's already over 600 sources of different data. And you can start to see what that data is in terms of the, it tells you about the data, so the metadata. You can then see how you would access that data. For some examples, you can see some of the research going on that data. You can see the types of publications being coming out of that. And also it's got a range of different training tools and educational courses associated with it. And this is now widely used. So you can see this, the monthly searches, so there's about 12,000 monthly searches going on that. So it's getting the sense of this growing use of different health data sources for research that's going on. So I mentioned earlier on about COVID and what's happening with that. For Health Data Research UK, this has been really quite an extraordinary year. So when COVID first kind of happened and the pandemic started in the UK, we were immediately having to respond to that and doing a number of things. So one of them was ensuring that that really the research was being done in a coordinated way. If you can imagine the amount, and all of us are now, from having heard over the news over the last year, the amount of research that's been going on, that needed to be coordinated, to organised, to making sure that the most urgent things were being prioritised first. We're then having to make sure that the access to the data that was needed was happening in an accelerated way, so that we really were getting the research happening quickly on there. And then the aspects of actually ensuring that the work, the health data science capability was really addressing the wider issues. So not just the immediate direct impacts, but those more indirect impacts, such as the impacts on delays to various treatments in cancer or cardiovascular disease. So we're really, and the impacts on different groups of population. So this is really how the data could be used in a much wider and broader way. So as one part of how we've been doing this is reporting to SAGE on a fortnightly basis. So these are little snapshots of the reporting we provide, and it's all available online. So you can look back over, it's quite a fascinating history of the evolution of the pandemic, actually. So what we've been doing on a fortnightly basis is showing the research that's been going on using health data. And it started off with very kind of nascent things, and then it's evolved into a really broad range of different researchers that have come off using the data. It also shows the status of the data, the new data sets coming on board, the new linkages being made to the data, and actually how accessible the data has become for different purposes. So we've been reporting this through to SAGE so that they can see actually how things are progressing and where things need to be supported and accelerated further. And as also as part of this, working with Patrick Vallance is something called the National Core Study. So this is a program of research that's evolved really over the last, since last summer, of bringing together in a really coordinated way the big national research programs, so that each of these is happening at the scale and the pace and the investment needed to make it happen across the UK. And you can see from the titles here, you've got kind of activities that Ian Diamond from Office of National Statistics is doing on surveillance and epidemiology. There's longitudinal health and well-being, the various clinical trials activities, and also then Andrew from the HSC on the transmission environment work, and then Paul Moss over in Birmingham looking at the immunity side of the disease. And then we're working across all of this in the data and connectivity piece, working with the Office of National Statistics to ensure the data is accessible and usable for all these different purposes. And so this has been creating a huge program of work of making sure all of that data is available and accessible for the right purposes in a trustworthy way. And we've been doing this in very much in close partnership with the public, and it's been absolutely fantastic through the whole period of the responsiveness that people have had to being helping and supporting in all sorts of really practical ways, ranging from responding to priorities on research questions, to helping design the ways in which members of the public are engaged in different clinical trials. So really practical ways of building that the use of the data, so that people can be confident that the data is being used for appropriate purposes and for the public benefit, but also that it's being made accessible quickly to answer the urgent questions. So the types of things that have been coming out on through this. So as I mentioned, the hubs earlier on. Now these were set up quite amazingly, really just a few months, 2 or 3 months before the pandemic started. And so they've been very much shaped through this year in terms of what's happening. It was fantastic to have that capability within the UK. And you can see from the range of things that they've been doing. So for example, DigiTrials was a key part to the recovery trial, which was then discovered the benefits of dexamethasone. We had Pioneer was working in looking at some things working very closely in the acute setting, and discovered guidance for VTE which is now being used across 60 countries. So really interesting kind of work and applications of using the data. There are also, you can see from this, the extraordinary number of people who've been involved in this work. So it's over 16,000 contacts with patients in the public to support this sort of work on there. And so what I'm going to do, the kind of last part of what I'm going to share is a couple of case studies that show really the practical use of how the data is actually being used. So this first one is, I mentioned the recovery trial, but the principal trial is another example. So the principal trial was actually set up for really to look for therapeutics or treatments that work for COVID for older people but in a community setting. So recovery is for people within the in hospital. This is for people who've got COVID, but before they go to hospital, are there treatments that could help them at that point? And one of the key challenges with that was actually finding people who've got COVID and then getting them onto the trial in the time frame before they either get better or they unfortunately get worse and need to be hospitalized. So this is very tricky to find that kind of group of people. And by using data, we were able to work with this is working with NHS DigiTrials to be able to actually use the testing data to identify and approach people who'd be suitable and offer them the opportunity to be involved in the trial once they've tested positively. And this meant that going from prior to doing this, there were just under 1,000 people had been recruited to the trial, and now up to nearly 5,000 people. So really quite a dramatic change, which then makes a huge difference to the effectiveness of what the trial can do and what it can be used for. So a really nice example of quite a simple application of using health data, making a big difference very rapidly during the COVID period. And the second one was then to another example of really a slightly different one. Then many of you will recall back in mid-February when we first heard the results of the vaccine effectiveness and from up in Scotland. What you may not have been aware of was the data that needed to be pulled together to do that. So this is talking through the EVE2 data set, which was the Breathe Hub was enabling. And what they were able to do by bringing together the vaccination data, the data in primary care setting, testing data, hospitalisation data, kind of various outcomes, able to then see the impacts of people of the vaccines. And from doing this, we were able to see the kind of the reduction in hospital admissions. And what was so important about this was the timeliness, the speed of doing it meant that this enabled the kind of building the confidence of not just the kind of political confidence, but also from members of the public confidence in the effectiveness of the vaccines. So really kind of very kind of important, two really important applications of using health data and how it's helped all of us in the last few weeks and months. And so the final thing I just wanted to touch on was a reflection on just looking back over this, how far we've got through the COVID experience is really, the first lesson is really around data being an absolutely fundamental resource to helping save lives and improve lives. And then the second one but doing that has required all sorts of people working together with different skills and different experiences. So it really is a collaborative, endemic effort to make this happen. And then the final lesson really is then the members of the public, patients, are really important enablers of using health data and important advocates for why this matters and why it's so important. So on that note, I shall hand back to Richard, and I'm looking forward to hearing the presentations from Angela, Adam, and Ewan.
Thanks, Caroline. That's a really excellent overview of the really important work that HCI-UK has been involved with on COVID-19. So thank you. So we're going to now shift gears slightly and focus on some research examples of COVID-19 research projects being conducted from HDR-UK Cambridge. I'm delighted to welcome 3 senior researchers and leaders from across the University of Cambridge and the Wellcome Sanger Institute. So firstly, we'll have Dr. Angela Wood, who's a reader in health data science in the Department of Public Health and Primary Care at the University of Cambridge, who will present a national cohort for COVID-19 research. Next, we'll have Dr. Adam Butterworth, a reader in molecular epidemiology from the same department as Angela. And Adam will be speaking about prioritising drugs for COVID-19. And last but not least, we'll have Ewan Harrison, who is a research fellow from the Wellcome Sanger Institute, who will present the COVID-19 Genomics Consortium. So just a reminder to put any questions you might have in the Q&A function, which you can find at the bottom of your screen, and we'll come to them at the end. So with that, I will hand over to Angela.
Thank you, Richard, and thank you to Caroline for really setting the scene and putting the importance of actually the public at the forefront of this. I'm really excited about this opportunity this evening to present to you. As Richard just said, I'm going to present on a new electronic health record resource on the whole population of England, which we can now access for research. Now, at the start of the COVID-19 pandemic, as a researcher, we had no access to healthcare data across the whole population of England to enable critical research to inform public health decisions. Now, there were 2 reasons for this. First, there was no collection of records from all the primary care doctors in England. Instead, the data collections were held by different computer system suppliers used by different general practices. And then second, there was no secure mechanism for us researchers to conduct the research using data from across the whole healthcare system. So not just from primary care, but from hospitals, death registries, and COVID test data. Thus, it was critical to establish a way in which we could securely access health data in England for research while maintaining public trust. So the next slide, please. Thank you. So the work was motivated by the need to increase our understanding of the relationship between COVID and cardiovascular disease or heart disease. And so the new resource was established by what we call the CVD-COVID-UK Consortium. This is a HDR-UK project that Caroline just mentioned, and it was under the leadership of the British Heart Foundation Data Science Centre and partnership with NHS Digital. Now, the resource comprises of person-level electronic health records from, and it's in this middle box here, from primary care, from hospital episodes, death registries, dispensing drugs and treatments, and COVID-19 test data from Public Health England. And plans are in place to further enrich this data by adding specialist intensive care data, cardiovascular registries, and also COVID-19 vaccination data. Now, all of these separate data sets come from different healthcare settings, and they've been linked by the NHS Digital team and have been put into a new trusted research environment so that researchers can have access to what we'd call de-identified data. So we can never see people's names or addresses or actual birth dates. So it's all de-identified. And as researchers, we go into this system. We can't actually pull data out or individual data records. It's a very secure environment hosted by NHS Digital. And over the next few slides for the remainder of this talk, I'm going to summarise the key features of this resource through describing a cohort of 54.4 million people who we were able to identify were alive at the start of next year. So that's around 96% of England. Thank you. So characteristics of the data of these people are shown on this slide here, and they match, as you would expect, the population of England. Around 51% of us in England are female, and 14% are age 70 or over. And there's a mean age of females slightly older than men of around 41.5 years, and for men at 40 years. Now, a really important feature of the data, because what's happened is it's been linked, people's electronic health records, say from primary care and hospital admissions and death registrations, they've all been linked by NHS numbers. And it means that we can gather more information. So this is just an example here of this Venn diagram showing how we can get the information on ethnicity, which is really important for our analysis. And you can see that around, oops, hang on, it's a bit small on my screen, but over 80%, actually I really can't see, I just need to make it a little bit bigger. Yeah, that's fine. So 63% of records in primary care recorded information about ethnicity, and over 80% recorded it on ethnicity from hospital admissions. And by pulling it together, pulling these two resources together, we get around, well, we get 51.5 million records on ethnicity, which is wonderful. Next slide, please. Thank you. And so here we show further this importance of linking data from different healthcare settings. And here we're counting cardiovascular events during the period from the 1st of January to the 31st of October last year. And what we're looking to do is kind of count the number of first ever, say, stroke or transient ischemic attacks. So these are kind of mini strokes amongst people who've never had them before. So that's why we're saying first ever. So we exclude people who we know have got a prior diagnosis, and we're looking to see how many people had these new events last year. And you can see the data is recorded from primary care, from hospital admissions, and the death registries. Now, importantly, we can see that actually, although some of the data overlaps, sometimes it's only recorded in primary care. So here we see 30% were only recorded in primary care, and only 4% in the death registrations. Now, what's nice is we can compare this with other diseases such as myocardial infarction or heart attacks. And here we can kind of see that the construction of where the data comes from is a little bit different. So here we see that 8% were only ever recorded in primary care. And that's because heart attacks are more of a, need immediate medical attention. And so many are rushed through into hospitals, whereas TIAs and mini strokes are often only ever recorded in primary care. And you can see that there's a higher proportion also of heart attacks that are only ever recorded on the death registrations. Now, this was last year. Sorry, just go back one more slide. Thank you. This was last year. And what we plan to do is investigate how these proportions from 2020 differ from previous pre-pandemic years as well. So next slide now. Thank you. And among this linked cohort, we identified nearly 1 million people with a confirmed or a suspected COVID-19 diagnosis. And this concords with the public health official statistics that have been reported as well. And while there's around 58% of diagnosis that have been recorded in both primary care and the COVID-19 lab test data, we can see that still there were 15% that were only recorded in primary care and not coming from the lab tests. Now, the reason for this is that actually the lab test data only ever is the definite positive lab tests, whereas in primary care, there are broader definitions that can be used, which includes a clinical or epidemiological diagnosis of COVID, but without a confirmed test result. And this is especially important to use this information from the start of the pandemic, where not everyone was going and getting tests. Okay, next slide, thank you. And on this slide, what we're showing is how the COVID-19 diagnosis reported from the different healthcare settings by sex, and then age, and then by ethnicity here at the bottom. Now, each bar is representing the percentage of the defined group with a COVID-19 diagnosis reported in the different healthcare settings. So, for example, there's around 1.5% of females had a COVID-19 diagnosis from the lab tests, compared with 1.3% of males. So whereas females are slightly more likely to have a COVID-19 diagnosis in their primary care and their COVID-19 lab tests, actually we see the reverse is true when we start looking at records in hospital and on death records, where we see that it's slightly more males. So for example, I think it's 0.1% males here in the death certificates compared to 0.08% of females. And in a similar fashion, we see something for older patients as well. So it's those under 70 that are more likely to have COVID-19 diagnosis in primary care and COVID test data, but it's the older individuals who are much more likely to have those reported in the hospital and the death registrations, which are much more likely to be severe COVID events. Going down to the ethnicity plot at the bottom, we also see a higher proportion of Asian and Asian British people have a COVID-19 diagnosis in the primary care and in the COVID-19 lab test data in comparison with other ethnicities. However, such differences are not observed in the records from hospitals or in death certificates. So importantly, it's very likely that the differences in the age distributions between the ethnic groups are leading to some of these results that we see here as well. Next slide, please. Thank you. So one of the key benefits of conducting research on whole country cohorts includes our ability to look at a wider spectrum of scientific questions within minority groups and disease subtypes. So here what we're showing are people with a previous history of stroke or TIA or mini stroke. They're much more likely to have a COVID-19 diagnosis recorded in all healthcare settings. So these orange bars are much taller than the blue ones, and more comparatively higher in hospital admissions and death registrations. Now, as previously, each bar is representing this proportion of the defined group with a COVID-19 diagnosis reported in that healthcare setting. So, for example, we can see that there's, hang on, if you just go on to the next slide. Thank you, brilliant, that's great. On this slide, what we now start seeing is also individuals with a previous history of myocardial infarction or heart attacks, obesity and diabetes. And actually we see this very similar pattern of individuals with history being much more likely to get COVID positive tests, both in primary care, lab tests, and hospital admissions and death registrations. Okay, you can move on to the final slide now. So this is just a summary of what I've just presented. So this is the first ever provision for research of linked national electronic healthcare data from across 96% of the population in England. The resource provides linked person-level data from across the different healthcare settings, which is really important for being able to define different cardiovascular disease outcomes, defining COVID-19 positive diagnosis and suspected diagnosis, and maximising the completeness of key characteristics such as ethnicity. And by including data from almost the entire population of England, the resource then becomes inclusive of all age groups and of ethnicities, geographical locations, and socioeconomic, health and personal characteristics, and thus can really enable powerful statistical research based on very large numbers of peoples and outcomes of interest. Now, of course, I said right at the start that the resource was initially set up to support research on COVID-19 and cardiovascular disease, but it will benefit other future initiatives as well. And just one final shout out to the fact that the resource is accessible by approved researchers through a very secure, trusted research environment by NHS Digital. Yeah, you can go to the final slide now. And so as Caroline alluded to in her talk, this endeavour has been part of a really large team, and what I'm showing here are just some key people involved in that. And so it's a cross-institute research team from the University of Bristol, UCL and Edinburgh, and others as well. And I should say a really strong public and patient advisory panel that really guide us to make sure that we're always answering the key questions of interest. And I've already mentioned the NHS Digital team, who have been a pleasure to partner with on this endeavour. Thank you very much. I think I'm now passing on to Adam for his talk, and we'll take questions at the end.
Thanks very much, Angela. It's a remarkable resource to have 96% of people in England in that database that can be analysed. I'm going to switch gears slightly, as the title suggests here, and tell you a little bit about a project that we've done over the last 12 months to help try and prioritise drugs that could be used for COVID-19. Caroline mentioned some of the trials that the UK has played a leading role in, such as Recovery and Principle. And one of the challenges for these trials is working out which of the many treatments that have been suggested could work for COVID-19 to test in these trials. There's only a select number of drugs that one can feasibly test. How can we prioritize which ones we might like to put into these trials? And this was an international collaboration, and you saw on the first slide here some of the collaborators around the world, particularly in the middle, Liam Gaziano, international joint PhD student between Cambridge in the UK and Cambridge and Boston in the US, who's really driven this work. So if you go into the next slide, the starting point for this was a project that we kicked off about a year ago, funded by HDO UK, that I lead, which is called the Multi-Omics Cohorts Consortium. The idea here is to bring together several of the cohorts from around the country, and you can see on the map on the left in the red dots, the location of some of the coordinating centres of these big population cohorts. And between them, they encompass something like 800,000 participants in these cohort studies. And the idea is that they share common features, and that working together, we could produce much better science than we could do working individually. So our aim is to really integrate different kinds of data that we have at large scale, and that includes genetic and genomic data. It includes health data of the types that Angela was talking about, linkages to health records. And it also includes what we call multi-omics, and that means other things that we can measure in the blood, like protein levels or gene levels or metabolite levels, for example. And there's a series of different aims that we have on the right-hand side here by building this consortium. And today I'm going to focus on the fourth one at the bottom, about how we can inform development of medicines, or in this case, perhaps prioritization of which medicines to test in trials. So on to the next slide, please. As I say, we kicked off this project almost exactly a year ago today, at the start of April. And when we put in the pitch for this project, we were thinking about testing our ideas on diseases like cardiovascular disease and cancers. And of course, at the time, nobody had heard of COVID-19. But as the project kicked off, we thought, how can we use the health data and the kinds of cohorts and the kinds of information we have to do something to help make a difference? On the next slide, you see one of the things that we did. Many of the cohorts around the UK that are involved in the consortium were able to link the participants who were in their own cohort with the kinds of data sets that Angela was talking about, particularly, for example, hospital data to say who'd been hospitalised with a COVID diagnosis, and also test data from Public Health England. So which of the participants in the cohorts had had a positive or a negative test. We contributed our data to a truly global initiative and international collaboration, nearly 50 studies now, 19 different countries. And you can see in panel D in the bottom middle here, quite a number of different cohorts from across the UK who've been able to contribute to this. If you go to the next slide, you'll see that this collaboration has now produced some results. What you're looking at here is a map of the human genome, ranging from chromosome 1 on the left over to chromosome 22 and the X chromosome on the right. And on the vertical axis, we have a measure of signal strength for each of the millions of genetic variants that we were able to test in this analysis. And you can see that there are about a dozen or so of these kind of yellowy stacks that rise up, which are regions of the genome that seem to have a strong statistical association with whether or not you end up being hospitalized for COVID-19. This is really exciting. It showed perhaps something we didn't know beforehand, which was, is there any component in our DNA which affects our likelihood of experiencing COVID-19 in a severe way? But actually, these are relatively weak signals. We can't really use these very well at the moment to predict who might or might not suffer from COVID-19 or suffer severely. One of the other challenges is here I've listed the names of these genetic variants, these RSIDs with long strings of numbers. But actually turning that into biological information, working out what are the key genes or proteins involved is another challenge. So if you go into the next slide, please, we were then thinking within our consortium about how can we use the kinds of health and molecular data we have to work out which drugs might help treat COVID-19? Can we suggest some candidates that could be tested in the kinds of national and international trials that Caroline mentioned? So here's how we did it. Here's one approach that we use. If you go back there, yeah, thank you. On the left of the slide is the gold standard. What we'd ideally like to do is this kind of randomized trial idea. And Caroline mentioned the world's earliest randomized trial several decades ago. But of course, this is what we use now to work out whether these drugs work. So we take a sample of people and we randomize them either to get the drug or usually a placebo. And then we might see, for example, if the protein that we're targeting, do the levels go lower? And does the disease risk or the outcome we're interested in get lower? And hopefully in the placebo group, we see the opposite. Those people have higher levels of protein and higher risk of disease. What we're trying to do is say, we would like to do the trial, but at the moment we need to first prioritize. We need to work out which drugs could be good targets. And the way we do that is a process called Mendelian randomization, sometimes known as nature's randomized trial. And this time, rather than randomizing people ourselves to get a drug or placebo, we exploit the fact that alleles are randomly allocated from parents to offspring through the population. And so some people will inherit a genotype at a particular variant, which gives them slightly lower levels of a protein, and other people will inherit genetic alleles that give them slightly higher levels of a protein. And we can then see if we think that that particular genetic variant is very specific to the levels of that protein, we can then see, do those people get higher or lower disease risk? So it's sort of analogous to the randomized trial on the left, but this is much quicker. We don't have to wait for the trial. We don't have to recruit patients. We can use data we've already generated. And if you click on one, I tend to think about this as on the next slide as the drugs have very powerful effects. They're the kind of sledgehammer approach. What we're looking for is subtle DNA variation that's like dimmer switches. It's turning things up or down slightly. And hopefully we can find lots of these to see in aggregate, if we turn all of the dimmer switches up a bit, do people have higher risk of disease? If we turn them all down a bit, do people have lower risk of disease? And that might inform us about what the sledgehammer approach might do. So what did we do? Well, one of the problems we had first is that if we were doing this for heart disease, a disease that's been around for decades and is likely to be around for decades, we can afford to identify new drug targets. Here with COVID, we had to move quickly. So the first thing we did is we worked with partners from the European Bioinformatics Institute to say, out of all the 20,000 human genes, which ones do we think could be very rapidly actionable? So there's maybe already a drug that targets them that's either on the market, it's being used in patients, or it's in development at the moment. And this gave us about 1,200. We then brought together some of our cohort data, for example, the interval study, which is a study of blood donors across England, which is coordinated from Cambridge. And we had measurements in nearly 5,000 people of about 350 proteins. We also worked with the Fenland study, another one in our consortium, again, coordinated from Cambridge with 10,000 participants this time. And we also accessed data that were publicly available from the so-called GTEx consortium from about 15,000 genes in about 600 people. And by basically bringing these data together, we could try and do a very large scale Mendelian randomization study testing these 1,200 actionable proteins to see which ones might be causally related to COVID-19. Luckily, we also found some signals, and you might be unsurprised to see that some of them lie in the same places as the stacks we saw previously. We of course wondered whether some of these were plausible and useful, and I've highlighted two examples here on the right. So in the bottom right, you see a protein called ACE2. This was immediately reassuring, the fact that this one came up because ACE2 had been discovered in the early stages of the COVID-19 pandemic to be the receptor that allows the virus SARS-CoV-2 to enter into the cells. So that was reassuring that we saw that as one of the things that came out of this very wide open experiment. The other one that we established was particularly interesting is this one called IFNAR2. This is a receptor for a set of proteins called type 1 interferons. This made a lot of sense biologically because these proteins relate to immunity, immune response. And it was also promising because there are drugs that act on this pathway that are already used to treat several diseases such as multiple sclerosis. And so this immediately suggested that these kind of drugs could be useful as treatments for COVID-19 patients. So I think looking at my summary slide, it gives a lot of the same messages that we've seen already from Caroline and Angela, really that national and international collaboration, if we can bring together these relevant health data sets and importantly, the different expertise that we had from around the world, we can really try and answer important questions. In this particular case, by the fact that we had genetic data, we had protein and gene expression data, as well as the health outcome data, we could try and identify these pathways that we think are important in how people respond to COVID-19, and that this could be used as a way to rapidly prioritize which drugs we might want to test. Perhaps slightly surprisingly to me, at least, the methods that we were used to using on diseases like cancer and heart disease also seem to work on infectious disease. And really, if we've learned anything in the last year, it's about how high quality science can really happen at high speed when suddenly everyone pulls in the same direction with very high urgency. And this project, which is going to be published in the next few weeks, has been a great example of that. And so on the final slide, just to reiterate that this was really global team science, and you see here the list of several dozen people who were involved to make this project happen from several different countries and multiple continents. And I'll hand over now to Ewan, who's going to tell you a bit about the COG-UK Consortium.
Hi, thanks. Can we go on to the next slide, please? So the COG-UK, or the COVID-19 Genomics Consortium, was stood up approximately this time last year, by a group of academic scientists who realised that the UK's genomics capacity could be leveraged to track SARS-CoV-2 in the UK. And the UK's got quite a strong track record in pathogen genomics all across the country. So COG-UK is made up of 16 academic institutions and the UK public health agencies. And also we have a large sequencing site at the Wellcome Sanger Institute just outside Cambridge. Each of the 4 UK public health agencies are also part of the consortium, and we were very lucky that very early on in the pandemic we submitted a funding application to government and received £20 million in funding, which allowed us to basically stand this up within a matter of days. going from the kind of first plans in early March, genomes were uploaded, and the first computational infrastructure that was required to analyze the data was stood up within a matter of 10 days. So really the ability to get on with it, because we had the funding, was absolutely critical. And the overall consortium is managed out of the University of Cambridge and is led by Professor Sharon Peacock, who is a professor of clinical microbiology within the university. So if we go on to the next slide, please. So how do we work? Well, we take the leftovers of diagnostic tests. So that can either be from the UK Lighthouse labs. So these are the big diagnostic testing facilities all across that take, if you call up with COVID-19 symptoms and you go drive to a car park or you send in a home test, they will end up in one of the large UK Lighthouse labs, which can process up to 20,000 samples a day. If you end up in hospital, we also take your samples if you're really poorly from COVID-19 disease. And our samples are split basically between two different pathways. If they come from the Lighthouse labs, in general, they end up at the Sanger Institute outside Cambridge, and they're dealing with over 500,000 samples a week, and they have to cherry pick them down from plates that contain thousands of both positive and negative samples before they can be sequenced. And if your sample comes through the NHS, it likely would have come through one of our 16 regional sequencing sites all across the UK. All the data is generated, sorry, just go back a slide. All the data is generated and flows into a central computational system that was already funded by the MRC and has now been expanded. And this was a critical component of our ability to stand up. The national consortium was existing UK infrastructure that was really amenable to data analytics. The data from patient information also flows into there, and I'll go into a little more detail about that. And then we have standardized analytical quality control and analysis pathways that mean then the data flows back to the UK public health agencies, back into NHS laboratories, but also back into the whole research network, which Health Data Research UK is a part of. Can we move on to the next slide, please? So one of the things that we've done in terms of data, from day one, the COG consortium decided that we should make data publicly available. So all our sequencing data is made available. Basically from, it gets sequenced, the sample gets sequenced within a matter of days. It's made available. First of all, we just put it on our website. It's also now available in a number of different archives. So there's one called GISAID, and also into the European Nucleotide Archive and other databases globally. We make all our data actually available. You can follow a link here on our website, which allows you to view the data in terms of phylogenetic trees and location data. We also make all our kind of reports and other things that we've developed freely available. And this has been really critical to the international effort of sequencing, that we've used this as leverage to ensure that other countries are also making their data really publicly available, because that's a critical part of being able to respond using genomics to understand how the virus is spreading. Can we go to the next slide, please? So within our consortium, so this really focuses on the kind of work that Health Data Research UK and has been doing, and staff from the Cambridge site have been critically involved in this from day one. As I mentioned, all our data flows into a single computational system called COVID-CLIME, as well as we kind of have three levels of data that we can hold. The first one is the public data that, as I mentioned, that we make publicly available. So that's just basically the sequencing data of just the virus. Any information other than the virus is removed. And we also release the data the sample was taken and the UK nation. And we've advocated this approach to ensure that data is publicly available for people around the world to use. But also there's no risk of deductive disclosure, so people understanding something about an individual from this data, because at the level of the UK nation and the sampling date, that information is not identifiable. We also take further steps if for example, the Scottish Islands, where cases may well be quite rare, we amalgamate that data even at a higher level to prevent identification. Within the consortium, we have access to more data that comes via the public health agencies. So we have very basic information about outer postcode, age and sex. We combine that with the genomics to track the virus as it moves across the country, to understand pathways, to understand introductions. And we can hold more restricted data, which tells us things more about hospital outcomes, etc. Can we move on to the next slide, please? So how do we use this? Why is data integration really important? So one of the things that we've done since the beginning of the pandemic is really to think about how we can integrate our data into other existing cohorts of data to answer important questions about COVID-19 disease. So one of the things that we've been doing really since early on is trying to ensure we sequence cases from severe COVID-19 disease so that the data can then be combined with things about comorbidities, outcomes, to understand how the virus itself and mutations in the virus may impact on outcomes of COVID-19 disease. We've linked up with a study called the genomic study, which is looking at host genes and mutations in our genes that may cause increased susceptibility to severe COVID-19 disease. And they've identified a number of novel genes that have been involved. We've also most recently, and I'll come on and talk about this, that you may have heard the variants of concern. So these are the particular lineages of the virus that have emerged really since in the last 3 to 4 months. And we've captured data and linked cases to those. And again, I'll talk a little bit more about how that's important. We're also looking at immunity. So we're sequencing cases from identifying, we need to identify which individuals have had a vaccine. So there's big data flows that are involved in ensuring that we can capture, who of us have had a vaccine, if someone's been previously infected. These are all critical questions, and data linkage is absolutely critical to answer those questions. We've also been looking at transmission in hospitals. We have a number of studies that have looked at transmission in hospitals and care homes. Again, data linkage is really critical to this. And we've also been looking at, you may have heard of the ONS studies. So these random samplings of the population each week to identify how many of us have COVID-19 disease, and also how many of us have antibodies to SARS-CoV-2. We're also again linked up with data for those to ensure that we can bring them together. So how do we do this? Well, we've had to come up with a unique way to do this, because we've actually managed within our data set to bring together data across the UK, so for all 4 nations. And as I said, we make our data publicly available, but for reasons of patient confidentiality, the linkage that we hold, so that is a sequence that links to an individual patient, that can only be held by the UK public health agencies. So to ensure that this data can flow out into other areas where there's really quite detailed clinical records that are held, so Health Data Research UK has been really critical to this, and we've built a system that will allow linkage of our public data to patient records that are held in what are called trusted UK research environments. So you may have heard of this one called SAIL in Wales, the Scottish Safe Haven, also Genomics England hold one, and NHS Digital. So what we do is we flow the viral sequencing data into these trusted research environments, where there it can be linked anonymously with complex both demographic but also electronic health data. And I'll just give you a couple of examples now of how this has been put to use already to understand things about COVID-19 disease. So early on in the pandemic, so this is, you can see here in the upper panel, we're talking about March, April, May last year, a mutation in the spike protein. So the spike protein on the surface of the virus enables it to bind to the ACE2 receptor that Adam mentioned previously on host cells. A mutation in this, a D614G mutation, was reported, and there were some papers suggesting that it had a role in increased clinical severity, but also in transmissibility. And our ability to link to these complex clinical data showed that actually, if you look in the bottom panel here, that there wasn't really any significant difference in the data set that we looked at in terms of the pathogenicity or clinical outcome of individuals with this virus. But we could model that there was an increase in transmissibility. So very, very quickly, because we had this data linkage, a new mutation occurred. There were experimental studies that suggested that it may be causing more severe disease. We could very, very quickly look in real world data to judge whether this or not was real. And finally, I think the best exemplar of how our data linkage has been used happened beginning in early December this year. So you may have heard of the Kent variant or the UK variant of SARS-CoV-2 that's spreading around the world. It's actually scientifically called the B117 lineage. And at the end of early December last year, cases in the southeast of England, so you can see here on the map where the darker purple shows, were starting to increase, whereas in the rest of the country there wasn't an increase in such a level. Because we have been conducting routine surveillance, so we're sequencing the virus day in, day out from across the country, we were able to go and look at these cases in the southeast of England and ask if there was anything particularly interesting about them. And what we identified, as you can see here on the right, this is a family tree of the virus, a phylogenetic tree, that there was a very long branch. So you can see that branch there with an arrow and a box showing you to it. And we identified that there was a particular lineage of this virus, now known as B177, that was transmitting within Kent in the southeast and London. And this particular lineage of the virus had 23 different mutations, which really made it stand out. So very quickly, we were able to use this data. We combined it with other data that was available from diagnostic tests, where one of the 3 targets of the diagnostic tests actually in this lineage wasn't working. So we could track this across the country, and we could see that this particular form of SARS-CoV-2 had been spreading across the country, and its prevalence were increasing. Now, what was critical here was that then again we could link with epidemiological information about the individuals, and we could compare virus transmission between people who had this particular lineage versus people who'd had other lineages of the virus. And this allowed us to very quickly demonstrate that the R, that's the transmissible, the reproductive number of the virus was higher for cases with B117 lineage, so that we could show that this was a more transmissible form. And more robust data has now shown that it's about 40% more transmissible. We were also able to, other researchers in the UK were able to link and look at severity of disease. So this was particularly people from the London School of Hygiene and Tropical Medicine. Again, by linking with this electronic health records, they could look to see whether it caused more severe disease. And again, they've shown that it has a moderate effect on severity of disease. So this data linkage work that have been going on behind the scenes, over the year, combined with the viral sequencing program that routinely sequenced virus from across the country, came together really perfectly in early December. And this allowed the UK government to, within a matter of days of this been identified, to inform the WHO and for countries around the world to close borders and take measures for this. And now we can see that this particular lineage has spread globally, but the data that was generated has obviously informed. We were prior warned about this happening. So I'd just like to on to the next slide. So I just want to finish on thanking I'm just a spokesman for the consortium. There's been over 600 people across the UK who've been involved, and 400 people who are working day in, day out to bring this consortium together. Thanks.
Thanks, Ewan, and thanks to all of you who've presented really excellent presentations that I think highlight how important it is to work collaboratively, nationally and internationally, and all the more impressive that all this work has been conducted, pivoting to this research within just the last year. So thanks a lot. We have a couple of questions, which I guess we've got a couple of minutes, and perhaps Angela, I'd ask you to come in on the first question here about people having died within 28 days of first positive COVID test, and how this affects the reliability of death data.
Yeah, it's a great question from Chloe Nicole. Thanks for asking that. And you're right, the way that it has been reported in the press, it has been about dying within 28 days of a first positive test. As researchers, we can be a bit more flexible, and we can kind of look at death certificates and what's been included on those death certificates. But we are moving to this place that if COVID is mentioned anywhere on the death certificate, or there has been a previous positive test, then we are counting that in the death toll, which I think is maybe a mistake. I know there's some really nice work being done from researchers in London in particular, where they're now really shifting and kind of thinking, actually, what's the excess number of deaths? So just as an example, somebody might go into hospital with a heart attack, they might get COVID in hospital, but then they might die, and COVID will be on their death certificate, but it's likely they might have died anyway. So it's kind of trying to detangle those as we do research. It really is a great question, Chloe, and I think we'll have to continue thinking how to do this to avoid any potential biases or misleading counts. And then the question from Brian about the GP records, and that actually you're absolutely correct, that the quality of the data starts right with the GP of how they've inputted the patient's data and whether it's been done correctly. I'm not a GP, but after speaking to GPs, I really do think a majority are very clued up on how they need to do this. And there's definitely a shift over time as well, where they realise the data is being used for research and not just their own records. And therefore it's that there's a much more greater appreciation of getting accurate codings which go in there. And kind of what happens is a GP puts in a code, and as researchers we use that code, but the way we'd interpret it is probably recorded diseases rather than absolute diseases, if that makes sense.
Thanks, Angela. That's very easy to understand answers. So that was great. Explained it well for me. Thank you. We've also had a question about funding cuts affecting free science research funding streams and whether this affects in HDR UK. So as far as I'm aware, it doesn't in the near term. Perhaps I'd ask Caroline to comment on this, but certainly not in the near term. Of course, the funding landscape has changed quite a lot recently due to COVID and other reasons. But Caroline, do you have any comments on that?
No, and I'm assuming this is referring to the overseas, the kind of ODA funds and things on exactly. That. So yes, you're right. It doesn't affect us directly at the moment, but it is obviously a very concerning in terms of the kind of signals and the impacts in terms of if grants and things are being cut and things being rescinded from here. That is obviously very concerning. We had conversations with the MRC around the impacts and of this, but at the moment it doesn't affect our current kind of funding profile, which is good from our perspective, but I'm sorry for everyone who is affected by it.
Yeah, and this alludes to the funding cuts from the overseas development aid budget from the government following the COVID-19 cuts to budgets. So I think that answers all of the questions. So it just remains to firstly thank all the speakers again, and to highlight what we've got on the screen now, that you can see more details about HDR UK at the link here. And for anyone who's interested in becoming a lay representative on our patient and public involvement panels, there's a link to the advisory group here where you can find details about that. And finally, just thanks everyone for filling in this final poll, and thank you very much again for attending. Thanks and goodbye.